Welcome to the tutorials on FSL-MRS, FSL's end-to-end spectroscopy analysis toolbox. In this course we have provided 3 videos covering 1. A very basic introduction to MRS, 2. An introduction to MRS preprocessing, and 3. An introduction to MRS fitting. Alongside these videos there is also a practical taking you through the analysis tools provided in FSL-MRS. In this first video, we will briefly walk you through some of the basic concepts around magnetic resonance spectroscopy and spectroscopic imaging. As you know, signal in MRI arises from the water molecules present in the body. In other talks, you've seen how just the signal from water gives rise to a wide variety of contrasts in MRI imaging. But to be more precise, it isn't the whole water molecule we are measuring. The signal arises, and the image is constructed from, the signal of 2 equivalent protons, or hydrogen atoms, on the water molecule. In spectroscopic terms, the signal from each of these protons occurs at the same frequency, and if we were to form a frequency spectrum, we'd see just one peak. This single frequency, at a known frequency, can be thought of as a metabolic fingerprint of water. But we know the brain contains many other chemicals of interest, neurotransmitters, energy substrates, and many metabolically active substances. Here we see just a few, and hopefully you will notice that they A. have different chemical structures, and B. because of that have many differently arranged hydrogen atoms. You can see a few of those protons here on glutamate. These hydrogen atoms, which inhabit different positions on the molecule, give rise to different signals in an MRI scanner. Thus, glutamate gives a unique spectrum or fingerprint, with particular peaks in that spectrum arising from particular collections of protons on the molecule. And each of the examples I'm showing here all have a different fingerprint, and in total there are about 19 different metabolites that we can observe in this way. When we collect a spectrum from the human brain, we observe all of the fingerprints summed together, weighted by the relative concentrations of those neurochemicals in the tissue we are imaging. By picking out the unique signals, we can then make images of the concentrations of the metabolites in vivo. These are known as metabolite maps, and here we see 3 such maps for total choline, glutamate, and NAA. So proton MRS can give us detailed information about metabolite concentrations in the target area. This, for example, can provide both sensitive phenotyping of diseases and monitoring of physiological changes arising from brain plasticity. But alongside this static metabolic profile, other types of spectroscopy are available. Recently, work combining proton MRS with diffusion is revealing cell-type specific measures of microstructure, or by looking at carbon atoms, phosphorus atoms, or using an exogenous tracer such as deuterium, we can measure dynamic aspects of metabolism. Metabolic fluxes can be measured through the oxidative phosphorylation pathway or the TCA cycle, or by using harmless deuterium labels simply ingested by the subject. The Warburg effect in cancer has been imaged. However, in this presentation we're going to focus almost completely on proton MRS. So in general, what substances are visible in proton MRS of the brain? Well, we should be able to see water. In fact, it should totally obscure the whole spectrum, as water has a concentration about 10,000 times larger than any of these metabolites. However, we take steps to deliberately suppress the signal from water. Strong lipid signals can also sometimes be seen. These can either be artifactual or present in certain pathology. We can then see certain metabolites with concentrations of around 1 millimolar or above, and this includes a subset of signals arising from the vast array of mobile amino acid chains and peptides in the body, and in MRS these are known as macromolecules. However, we can't detect very low concentration metabolites or solid substances, and this includes things bound to substrates or large relatively immobile molecules, such as whole proteins. A representative spectrum acquired from a volume of in vivo brain tissue is shown here, and this is actually quite a high-quality spectrum collected at 11.7 Tesla, but it gives us an idea of what we might be able to detect on a clinical scanner. We can measure up to about 19 different metabolites in a very tight range. We usually only look over this 1 to 4 ppm range, and ppm here stands for parts per million on the chemical shift axis, and that's a frequency axis which is invariant with field strength of magnet, i.e. it's the same at 1.5 Tesla as it is at 11.7 Tesla. I have to forgive the backwards axis, it's just a historic convention. With such an array of signals, it isn't surprising that the interpretation of information gained from MRS is complicated. And whilst many studies only focus on a few of the most prominent metabolites, the understanding of the uses and compartmentalization of those metabolites is still incomplete or disputed. Different brain regions, different subject age, and pathology is all likely to affect the interpretation of changes observed in spectra. Nevertheless, this slide gives a very brief summary of the most prominent metabolites in proton MRS, and some metabolites have very clear compartmentalization, such as NAA, which is almost entirely neuronal, but conversely, the oft-stated compartmentalization of myoinositol to glia is probably an oversimplification. Creatine and phosphocreatine, or total creatine, play a core role in the oxidative phosphorylation pathway, and by proton and phosphorus MRS we have a direct measure of that. However, the famous role of GABA and glutamate as synaptic neurotransmitters probably does not directly relate to the pools visible using MRS. And for the interested viewer, I direct you to these two resources for more information on this topic. So what equipment do you need to do spectroscopy? Well, luckily we can use the same scanners and the same hardware that is used for MR imaging. In general terms, the higher the field strength, the better for spectroscopy, as we both gain signal to noise, and the metabolic fingerprints don't overlap as much at higher field. However, over and above MRI, we need different sequences and some extra expertise in the operation of the scanner and in interpretation of the results. So let's now take a brief look at those special pulse sequences. To create an image using an MRI scanner, a sequence must be run to collect sufficient k-space information. And then we Fourier transform that to form an image. To collect a spectroscopic image, the same process must be followed, but in addition, an extra time dimension must be collected. This, in turn, is Fourier transformed to form our spectrum. As we've seen before, we can combine these two processes to create spectroscopic imaging and metabolite maps after analysis. And so we see that the collection of a time dimension is a key aspect of MRS sequences, and we'll now see two common ways that this is implemented. The first is single voxel spectroscopy, abbreviated to SVS. As the name implies, the sequence generates a single spectrum arising from one volume of tissue in the subject. That volume is about 2 to 3 centimeters square and takes around 5 minutes to acquire enough temporal averages for sufficient SNR. There are several common sequences in use, namely press, steam, and semi-laser, and each of them works by combining 3 orthogonal slice-selective pulses to restrict signal to a cuboidal 3D volume. On this slide you can see a schematic of a steam sequence, and you'll meet this again in the practicals. The second broad category of MRS sequence is MRSI. This might also be known as chemical shift imaging or CSI, and in MRSI spectra are simultaneously collected from many voxels, and the sequences are more akin to normal MR imaging sequences. Resolutions can be down to a few millimeters isotropic, but that does come with an associated increase in scan time, sometimes up to the tens of minutes. And examples include CSI or EPSI or more advanced KT-based trajectories like spirals or concentric ring sequences. MRS sequences must also contend with two additional challenges which otherwise will give rise to severe artifacts. The first is suppression of the dominating water signal. So several thousand times stronger than the signal from metabolites, the water signal, if not suppressed, will cause large baseline distortion to the spectrum. On the left, we see a figure which shows water suppressed and water unsuppressed signal from the same subject. And if we zoom into that 1 to 4 ppm range, we can see that metabolite signals do appear in each case. It's quite clear that the NAA signal appears in both at 2 ppm. However, in the case without water suppression, which is the blue line, there is clearly a large baseline distortion present. And we deal with this and we suppress this by adding a water suppression module at the start of our sequence. The second common artifact is that of lipid infiltration, arising from lipids in tissues surrounding the brain. Here we see 3 spectra acquired from 3 points in an MRSI scan. A normal spectrum is seen from the central region, but at the edge of the brain, the green spectrum, there's partial voluming with tissue outside the brain leads to a large distorting lipid resonance. However, even worse, at an intermediate point, towards the edge of the brain, but still clearly within the brain, some distortion is still present as a large lipid signal bleeds into nearby voxels. There are 2 ways of tackling this in a sequence. The first is to use the SVS localization technique to only collect signal from a region free of lipids. However, this restricts collection of cortical spectra close to the edge of the brain, which don't fit within that SVS volume. A second way is to use spatially selective saturation, usually multiple saturation bands, to suppress the signal arising from the edge of the brain. There's also data-driven post-processing techniques which can be used to remove lipids present in spectra that were already acquired. We'll finish this talk with a very short overview of the analysis of MRS data. For single voxel data, we can see here the subtle effects of pre-processing, and there's been several steps carried out. Coil combination is used to deal with phased array coils. Frequency and phase correction is then used to improve line widths. Eddy current correction is used to remove line shape distortions. And then finally, residual water is removed to reduce baseline distortion. And all of this will be presented in more detail in the following lecture. After pre-processing, fitting must be carried out. And we see here a short animation showing the process of fitting simulated metabolite fingerprints or basis spectra to an in vivo spectrum. This is followed by a process of quantification, referencing either to other metabolites or to the water signal to generate concentration in meaningful units. And we'll look in much more detail at the fitting process in the third video. So that concludes the first talk on MRS and FSL-MRS. Join us in the next video for a more in-depth look at pre-processing methods. But for now, I'll leave you with a few suggested resources on where you can get more information on MRS. See you next time.